Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
In the opening decades of the 20th century, the landscape of biology and medicine was haunted by phantoms. These were substances essential for life, whose absence caused catastrophic diseases like scurvy, beriberi, and pellagra, yet they remained chemically invisible. Investigators referred to them with a placeholder name, accessory food factors, or more evocatively, vitamins. Their existence was inferred entirely through their biological effects, a frustrating state of affairs for a scientific community increasingly grounded in the material certainty of chemistry. The central challenge was immense: to isolate these trace compounds from the bewildering complexity of natural foodstuffs and to determine their atomic architecture. It was a problem that demanded a new kind of chemical thinking, one that could bridge the gap between the predictable order of the mineral world and the sprawling, intricate chaos of organic life. Into this environment stepped Paul Kerr, a chemist whose intellectual pedigree seemed, at first glance, ill-suited for the task. His training was forged in the crucible of inorganic coordination chemistry under the tutelage of the formidable Alfred Werner at the University of Zurich. Werner's Nobel Prize-winning work in 1913 had imposed a breathtaking three-dimensional order upon the world of metal complexes, revealing elegant octahedral and square planar geometries where others had seemed only empirical formulas. This education instilled in Kerr a profound appreciation for stereochemistry, the precise spatial arrangement of atoms, and a rigorous, almost architectural approach to molecular structure. It was a mindset honed on platinum and cobalt compounds, a world of clean crystals and predictable valencies. Kerr's initial research after joining the faculty at Zurich reflected this unique background. He focused his efforts on complex organic compounds containing arsenic, developing new synthetic methods for molecules like arsenamine. This work served as a crucial intellectual bridge. These organoarsenic compounds were hybrids, possessing the structural complexity of organic molecules tethered to a metallic center. In mastering their synthesis and analysis, Kerr was sharpening the very tools he would soon need for a far greater challenge. He was learning to apply the geometric precision of Werner's world to the less rigid frameworks of carbon chemistry. This period was his apprenticeship in applying inorganic structural logic to organic problems, a preparation for his eventual pivot into the vibrant and mysterious world of natural pigments. The allure of these pigments was twofold. They were ubiquitous in the plant kingdom, and their brilliant colors, from the deep reds of tomatoes to the bright orange of carrots, suggested potent chemical reactivity. More importantly, a growing body of evidence hinted at a deep connection between these colorful compounds and the phantom vitamins. Richard Willstetter, a towering figure in German chemistry, had performed pioneering investigations into the structures of chlorophyll and the carotenoids, the family of yellow and orange pigments. Willstetter's work provided the first rough map of this new territory, yet his proposed structures for the carotenoids would ultimately prove incomplete. He had opened the door, revealing a tantalizing glimpse of a new chemical world, and it was Kerr who would step through it with a more refined set of analytical keys. Kerr chose to begin his exploration with lycopene, the pigment responsible for the rich red color of ripe tomatoes and watermelons. This was a strategic choice. The molecule was abundant, and its intense color made it easy to track during chemical reactions. Applying the classical techniques of organic analysis with unparalleled rigor, Kerr subjected lycopene to controlled oxidative degradation, using agents like ozone or potassium permanganate. He systematically shattered the long carbon chain of the molecule into smaller, more easily identifiable fragments. By painstakingly identifying these pieces, such as uronic acid, he could logically deduce how they must have been connected in the original structure. It was akin to reassembling a shattered vase by studying the shape of every shard. Through this meticulous work, published in the prestigious journal Helvetischer Chemiker, Kerr demonstrated that lycopene was a long, straight-chain hydrocarbon composed of 40 carbon atoms. He further established its fundamental relationship to carotene, the pigment from carrots, showing them to be isomers, molecules with the same chemical formula but different atomic arrangements. This investigation was a masterpiece of classical chemical elucidation. It showcased his signature method, a combination of precise degradation, comparative analysis of related molecules from different natural sources, and a deep, intuitive understanding of structural possibilities inherited from Werner. He was thinking about these large, flexible organic molecules with the same geometric discipline his mentor had applied to rigid metal complexes. The success of the lycopene work did more than solve the structure of a single pigment. It solidified the reputation of the Zurich School as a global center for natural product chemistry. Kerr's laboratory became a beacon for aspiring chemists eager to work at this exciting frontier. It attracted brilliant minds from across the world, including a young American student named George Wald. who would later build upon the foundations Kerr was laying. The methodological framework Kerr established with lycopene became the standard for the field. He had created a reliable blueprint for navigating the labyrinth of complex natural products. This foundational period positioned Kerr perfectly for the ultimate prize in the field. He had refined his techniques, built a world-class research group, and demonstrated his ability to solve the structures of large, unsaturated hydrocarbons. He had moved intellectually from the clear, geometric certainties of inorganic chemistry to the hybrid world of organometallics and into the rich domain of plant pigments. His work on lycopene provided the final piece of his intellectual armamentarium. Now, the scientific community was closing in on the identity of the vital fat-soluble factor A, essential for growth and vision, and its precursor, a substance found abundantly in carrots. The chemical mystery of beta-carotene and its relationship to vitamin A was the defining problem of the era, and Paul Kerr, the chemist trained in the logic of minerals, was uniquely equipped to unravel its organic secret. This foundational period positioned Kerr perfectly for the ultimate prize in the field. He had refined his techniques, built a world-class research group, and demonstrated his ability to solve the structures of large, unsaturated hydrocarbons. He had moved intellectually from the clear, geometric certainties of inorganic chemistry through the hybrid world of organometallics and into the rich domain of plant pigments. His work on lycopene provided the final piece of his intellectual armamentarium. Now, the scientific community was closing in on the identity of the vital fat-soluble factor A, essential for growth and vision, and its precursor, a substance found abundantly in carrots. The chemical mystery of beta-carotene and its relationship to vitamin A was the defining problem of the era, and Paul Kerr, the chemist trained in the logic of minerals, was uniquely equipped to unravel its organic secret. The challenge was immense, a true intellectual Everest for the organic chemists of the early 20th century. The biological effects of what was called vitamin A were well documented. Its absence led to debilitating conditions like cerophthalmia, a drying of the cornea, and stunted growth in the young. Conversely, its presence, often sourced from potent fish liver oils, restored health. Parallel observations, pioneered by researchers like Hans von Euler-Chelping, revealed that the vibrant orange pigment in carrots, beta-carotene, possessed the ability to act as a prototamin. Ingesting it allowed the body to produce vitamin A. This biological connection was a profound clue, yet it posed an even deeper chemical question. How were these two molecules related? What was a precise atomic architecture that allowed one to become the other? Answering this would require chemists who could think like a biologist and work like a master artisan. Kerr entered a field populated by brilliant minds, all racing toward the same goal. The work of contemporaries like Heinrich Otto Wieland on the intricate structures of bile acids had established a powerful precedent. It demonstrated that even the most complex products of nature could yield their secrets to systematic chemical degradation and logical deduction. Wieland's success created an atmosphere of ambitious possibility, providing a conceptual roadmap for dismantling large molecules to understand their construction. Kerr, having sharpened his own degradation techniques on plant pigments, saw the path forward. The problem of beta-carotene was a grander version of the puzzles he had already solved. It was a large, unwieldy, and notoriously unstable polyene, a long chain of carbon atoms linked by alternating single and double bonds. The key lay in finding a chemical scalpel precise enough to cut the molecule at a strategic point. His chosen instrument was ozone. The technique of ozonolysis, while known, was wielded by Kerr with exceptional finesse. Ozone attacks the carbon-carbon double bonds that form the backbone of the polyene chain, cleaving the molecule cleanly at those locations. Kerr hypothesized that if beta-carotene were a symmetrical molecule, as its biological role as a precursor to two potential vitamin A units suggested, then cleaving it at its central double bond should yield two identical, smaller molecules. This was a beautifully simple and elegant proposition, a direct test of the molecule's symmetry. The execution of this idea in his Zurich laboratory was a masterclass in experimental chemistry. Upon treating a solution of beta-carotene with ozone and then working up the products, Kerr's hypothesis was stunningly confirmed. The reaction yielded a single primary product, which he painstakingly isolated and characterized. He determined it to be an aldehyde containing a 20-carbon skeleton. Crucially, he then demonstrated that this very same aldehyde could be derived from vitamin A, known as retinol, which he had laboriously purified from fish liver oil. The aldehyde he had produced was retinol, and its corresponding alcohol was retinol, or vitamin A. The puzzle pieces snapped into place with resounding clarity. Beta-carotene was a 40-carbon symmetrical hydrocarbon. The body's enzymatic machinery simply cleaved it in half to produce two molecules of 20-carbon retinol, which was then reduced to form active vitamin A. This discovery, published as landmark 1931 paper in Helvetica Chemiker Actor, was a watershed moment. It provided the definitive structure of beta-carotene, A long polyene chain capped at both ends by characteristic beta-ion ring is simultaneously established the exact structure of vitamin A and explained their biochemical relationship with irrefutable chemical logic. To silence any remaining skepticism and to showcase the absolute power of this structural proof, Kara's group then achieved the first ever total synthesis of a vitamin. Starting from simple chemical precursors, they constructed the entire vitamin molecule in the laboratory, atom by atom. The synthetic vitamin was identical in every respect to the one found in nature. This feat erased any lingering doubt and firmly established the era of vitamin synthesis. The impact of this work radiated outward through biochemistry, medicine, and nutrition. For the first time, dietary recommendations could be based on a concrete molecular understanding. The fortification of foods with vitamin A or its beta-carotene precursor became a scientifically grounded public health strategy. Yet, the most profound influence was perhaps on the future of basic science. A young biochemist at Harvard, George Wald, was fascinated by the mechanics of vision. He knew from Kara's work the exact shape and chemical properties of retinol. Armed with his structural blueprint, Wald was able to ask a more refined question: what was this specific molecule doing in the eye? His subsequent research led to the discovery of the visual cycle, demonstrating that the isomerization of retinol, a subtle change in its shape upon absorbing light, is the primary chemical event of vision. Wald's discovery, which earned him the 1967 Nobel Prize in Physiology or Medicine, was built directly upon the chemical foundation Kara had laid decades earlier. One Nobel Prize in chemistry directly enabled another in medicine, a perfect illustration of the interconnectedness of scientific progress. Kara's triumph in the race for vitamin A showcased his defining strengths: a capacity for immense experimental labor, a genius for deductive reasoning from degradation products, and an unwavering focus on the most fundamental questions. He had conquered the world of long-chain aliphatic polyenes. His success, however, would have been a lesser achievement had he remained within this chemical comfort zone. The next great challenge in the vitamin field involved a completely different class of molecule, one with a complex, fused ring system and unique fluorescent properties. Having unraveled the secret of the linear carotenes, Kara would next turn his attention to the intricate heterocyclic world of the flavins, demonstrating a remarkable intellectual agility that would soon lead him to a second, equally significant, molecular prize. Kara's triumph in the race for vitamin A showcased his defining strengths: a capacity for immense experimental labor, a genius for deductive reasoning from degradation products, and an unwavering focus on the most fundamental questions. He had conquered the world of long-chain aliphatic polyenes. His success, however, would have been a lesser achievement had he remained within this chemical comfort zone. The next great challenge in the vitamin field involved a completely different class of molecule, one with a complex, fused ring system and unique fluorescent properties. Having unraveled the secret of the linear carotenes, Kara would next turn his attention to the intricate heterocyclic world of the flavins, demonstrating a remarkable intellectual agility that would soon lead him to a second, equally significant, molecular prize. The intellectual impetus for this new direction originated in the Berlin laboratory of the formidable biochemist Otto Warburg. In 1932, Warburg and his colleague Walter Christian had isolated a mysterious substance they called the yellow oxidation ferment from yeast. This enzyme was absolutely essential for cellular respiration, the process by which cells derive energy from nutrients. They managed to separate the ferment into two parts: a large protein component and a smaller, non-protein, colored prosthetic group. This smaller molecule was the active component, the coenzyme, and it possessed a striking yellow-green fluorescence under ultraviolet light. Its chemical identity, however, remained a complete enigma. The scientific community immediately recognized the profound importance of this discovery. Here was a key that could unlock the chemical machinery of life itself, yet the key's own structure was an unsolved puzzle. This puzzle presented the perfect challenge for Kara's Zurich laboratory. The problem required the same blend of large-scale isolation and meticulous chemical analysis that had proven so successful with the carotenes, yet it demanded mastery over an entirely different domain of organic chemistry. The world of fused nitrogen-containing rings was a far cry from the straight chains of vitamin A. Kara entered a fierce but collegial race with other leading chemists, most notably Richard Kuhn and Heidelberg, to isolate and identify this fluorescent coenzyme. The molecule's own brilliant fluorescence became the primary tool for the chase, a glowing beacon that allowed researchers to track its presence through countless purification steps. The task was immense. Kara's team processed hundreds of liters of whey, the watery byproduct of cheese making, and thousands of egg whites, laboriously concentrating the trace amounts of the yellow substance they initially termed lactoflavin. The isolation of a pure, crystalline sample was merely the first victory. The true intellectual battle lay in deciphering its molecular architecture. Kara deployed his signature strategy of controlled degradation, systematically breaking the unknown molecule into smaller, identifiable fragments. This was chemical archaeology, reconstructing a complex edifice from its constituent bricks. Through careful alkaline hydrolysis, his group cleaved the molecule into two principal components. One was a sugar alcohol they identified as a derivative of ribose, specifically a 5-carbon chain called ribitol. The other fragment was the core chromophore, the part of the molecule responsible for its color and fluorescence. To probe this core, Kerr employed a different technique, photolysis. By exposing the flavin to strong light, he induced a chemical reaction that cleanly severed the ribitol side chain, yielding a new, intensely fluorescent compound he named lumoflavin. The structure of lumoflavin was simpler to analyze, and through a series of further degradation reactions and elemental analysis, Kerr deduced its framework. He proposed it was a tricyclic system containing 3 fused rings with 4 nitrogen atoms, a structure known as an isoxazine. By piecing the evidence together, he concluded that the original vitamin, which the community was beginning to call riboflavin, consisted of this dimethyl isoxazine ring system with the ribitol side chain attached to one of its nitrogen atoms. This proposed structure was elegant, yet in the competitive world of chemistry, a proposal requires definitive proof. The ultimate confirmation of a molecular structure is its total synthesis from simple, known starting materials. In 1935, Kerr's group achieved this monumental feat. In a brilliant multi-step synthesis published in the journal Helvetica Chemicreactor, they constructed the complex isoxazine ring and then attached the ribitol side chain, producing a synthetic substance that was in every respect identical to the natural vitamin isolated from whey. It had the same melting point, the same elemental composition, and, most importantly, the same characteristic yellow-green fluorescence and biological activity. This synthesis was the final word in the structural debate. It confirmed Kerr's proposal beyond any doubt and corrected alternative structures that had been put forward by competing laboratories. The immediate impact of this work was profound. With the structure known and a synthetic route established, the industrial production of vitamin B2 became feasible. This allowed for its widespread use in medicine and food fortification, leading to the near eradication of deficiency syndromes like riboflavinosis, a condition characterized by skin lesions, inflammation of the mouth, and anemia. Kerr's chemical detective work had a direct and lasting benefit on public health. The deeper, long-term significance of his discovery, however, lies at the very heart of modern biochemistry. Kerr's structural elucidation of riboflavin provided the molecular foundation for understanding its central role in metabolism. Scientists soon discovered that riboflavin is the precursor for 2 of the most important coenzymes in all of biology, flavin mononucleotide, or FMN, and flavin adenine dinucleotide, or FAD. These molecules are the workhorses of cellular energy production. They function as essential electron carriers, accepting electrons from the breakdown of food molecules and shuttling them to the electron transport chain in mitochondria. This process, called oxidative phosphorylation, generates the vast majority of a cell's energy currency, adenosine triphosphate, or ATP. Every breath we take, every muscle we move, depends on the seamless functioning of this electron transport system, a system in which the flavin structure determined by Kerr plays an indispensable role. Kerr's achievement with riboflavin created a powerful intellectual lineage. His work stood on the shoulders of Otto Warburg, whose biological discovery provided the target, and it utilized the foundational principles of heterocyclic chemistry established by earlier giants like Emil Fischer. In turn, Kerr's success directly enabled the next generation of research. The British chemist Alexander Todd, who would later win his own Nobel Prize, embarked on the synthesis of the complete coenzyme molecules, including FAD and ATP. Todd's synthesis of flavin adenine dinucleotide was a direct extension of Kerr's work. Having the structure of the flavin component solved was the critical prerequisite for tackling the larger, more complex coenzyme. Having now charted the structures of 2 radically different vitamin classes, the aliphatic carotenes and the heterocyclic flavins, Kerr's laboratory had established an unparalleled reputation. The Zurich Institute became a global center for the chemistry of natural products. The systematic methodology of isolation, degradation, structural deduction, and definitive synthesis had proven its power twice over on 2 of the most important biological molecules of the era. With these triumphs secured, the logical course was to apply this formidable research engine to other vital nutritional factors whose chemical identities remained obscured in the complex mixtures of natural sources. The vast and varied world of lipids and other essential coenzyme components still held many secrets, presenting a new frontier for exploration. Having now charted the structures of 2 radically different vitamin classes, the aliphatic carotenes and the heterocyclic flavins, Kerr's laboratory had established an unparalleled reputation. The Zurich Institute became a global center for the chemistry of natural products. The systematic methodology of isolation, degradation, structural deduction, and definitive synthesis, had proven its power twice over on two of the most important biological molecules of the era. With these triumphs secured, the logical course was to apply this formidable research engine to other vital nutritional factors whose chemical identities remained obscured in the complex mixtures of natural sources. The vast and varied world of lipids and other essential coenzyme components still held many secrets, presenting a new frontier for exploration. The first of these new targets emerged from the field of reproductive biology. In 1922, the American researchers Herbert Evans and Catherine Bishop had identified a fat-soluble factor in vegetable oils, particularly wheat germ oil, that was essential for successful gestation in laboratory rats. Its absence led to fetal resorption, and they designated this substance vitamin E, the antisterility or fertility factor. For over a decade, the biological effect was well documented, yet the molecule responsible remained an elusive ghost within the oily extracts. The challenge was immense. The vitamin was present in trace amounts, and its lipid nature made it difficult to purify using the techniques of the day. This was precisely the kind of intricate structural puzzle that Kara's group was uniquely equipped to solve. His team initiated a painstaking process of extraction and fractional distillation of wheat germ oil, a task demanding immense patience and chemical finesse. They succeeded in isolating several closely related active compounds, which they named alpha-tocopherol, beta-tocopherol, and gamma-tocopherol, from the Greek words tokos for childbirth and pherin for to bear or carry. The isolation itself was a monumental achievement, but the true brilliance lay in the subsequent structural elucidation. Kara employed a strategy of controlled chemical demolition by subjecting alpha-tocopherol to vigorous oxidative degradation with reagents like chromic acid or nitric acid. His group cleaved the large molecule into smaller, more easily identifiable fragments. This chemical dissection yielded two principal components. One was a durable aromatic portion, a substituted chromane ring system they named trimethylhydroquinone. The other was a long, they named carbon aliphatic alcohol, which Kara's team correctly identified as vial, the same lipid tail found in chlorophyll. The puzzle pieces were on the table. Through meticulous analysis of the degradation products and clever chemical reasoning, they deduced how these pieces fit together. It proposed that the vitamin E molecule consisted of the chromane ring attached to the long vital side chain. This groundbreaking work culminated in a 1938 publication in Helvetica Chimica Actor that presented the correct structure of alpha-tocopherol, a finding confirmed almost simultaneously by its total synthesis in his laboratory. The elucidation of the structure had immediate and profound implications. It explained the vitamin's function at a chemical level. The phenolic hydroxyl group on the chromane ring was revealed as the active site, capable of donating a hydrogen atom to neutralize destructive free radicals. This established vitamin E as the body's primary lipid-soluble antioxidant, a guardian of cell membranes, protecting the delicate fatty acids within them from oxidative damage. This single discovery opened the door to the entire modern field of antioxidant biochemistry. The structural knowledge provided by Kara enabled subsequent generations of researchers, such as the pioneering lipid biochemist Al Tapel, to investigate the precise mechanisms of cellular protection and to explore the links between oxidative stress, aging, cardiovascular disease, and neurodegeneration. While the work on tocopherols was reaching its climax, Kara's systematic program continued on a parallel track, returning to the world of water-soluble B vitamins. The scientific community was intensely focused on pellagra, a devastating disease characterized by dermatitis, dementia, and diarrhea, which was rampant in populations with corn-based diets. It was known to be a nutritional deficiency, and a curative agent, the pellagra preventing factor, had been found in sources like yeast and liver. The work of previous giants, including Arthur Harden and Hans von Euler-Chelpin, had shown that a vital coenzyme, which they called casamase, was essential for fermentation. This coenzyme was later shown to contain the pellagra preventing factor. The challenge was to pinpoint the exact chemical identity of the active component within this larger coenzyme structure. The American biochemist Conrad L. Agin had demonstrated in 1937 that nicotinic acid in its amide, nicotinamide, could cure a canine disease analogous to pellagra. Kara's contribution was to provide the definitive chemical proof of this relationship within the coenzyme itself. He and his team undertook the synthesis of casamase, which we now know as nicotinamide adenine dinucleotide, or NAD+. By successfully building this complex molecule in a laboratory, he confirmed its structure showing unequivocally that nicotinamide was a key functional part of the coenzyme. His work demonstrated that the nicotinamide ring is the site of the molecule's action in biological redox reactions. It acts as an acceptor of electrons and protons during the metabolic breakdown of fuel molecules like glucose. This chemical conformation solidified nicotinamide's identity as vitamin B3 and explained its fundamental role in cellular energy production. A deficiency in this small, simple molecule crippled the entire metabolic engine of the cell, leading to the systemic collapse observed in pellagra. Kerr's synthesis provided the final, unassailable piece of evidence, connecting a dietary substance to a specific molecular mechanism at the very heart of life's chemical processes. The investigations into vitamin E and nicotinamide showcased the maturity and power of Kerr's research program in the late 1930s. He moved with confidence from one class of natural product to another, from a complex lipid-soluble antioxidant to a water-soluble coenzyme component. Each success was built upon a refined and proven methodology. He took biological observations made by others, like those of Evans and Bishop, and translated them into precise molecular structures. He took existing biochemical puzzles, like the composition of cosmase, and provided the ultimate resolution through chemical synthesis. This relentless drive to catalog and comprehend the essential molecules of nutrition was creating a comprehensive atlas of biochemistry. This impulse toward systematic organization and clear presentation of complex chemical knowledge would soon find its most enduring expression. Moving from the laboratory bench to the printed page, this transition from discoverer to synthesizer culminated in a work that would define the education of chemists for half a century. The explosive growth of organic chemistry in the early 20th century had created an intellectual paradox. Laboratories, Kerr's included, were producing a torrent of new data, isolating novel compounds, and elucidating reaction pathways at a breathtaking pace. Yet this wealth of information threatened to become a bewildering jungle for students and even seasoned researchers. The field was rich in detail, but impoverished in its overarching narrative. What was needed was a guiding hand, an authoritative voice to impose order on the creative chaos, to build a coherent intellectual edifice from the countless new molecular bricks being unearthed. In 1927, Paul Kerr provided that edifice. His magnum opus, the Leibach der Organischen Chemie, or Textbook of Organic Chemistry, was far more than a simple compilation of facts. It was a foundational act of intellectual architecture. While drawing from the esteemed German tradition of comprehensive chemical treatises, Kerr's book presented a revolutionary perspective. Previous textbooks tended to treat the chemistry of natural products as an exotic, almost separate discipline, an appendix to the more predictable world of synthetic compounds. Kerr obliterated this distinction. His textbook was the first to present the entire discipline as a unified whole, giving the complex structures of vitamins, carotenoids, alkaloids, and hormones equal prominence and systematic treatment alongside classical organic molecules. The genius of the Leibach lay in its elegant and logical structure. It organized the vast material primarily by functional groups, a pedagogical standard that persists to this day. Within this framework, however, it integrated deep, authoritative discussions of the most complex molecules known. A student learning about alcohols and ethers would also encounter the intricate chemistry of carbohydrates. A chapter on carboxylic acids would flow naturally into a discussion of amino acids and proteins. This approach had a profound effect. It taught generations of chemists to see the molecules of life, with all their staggering complexity, as extensions of fundamental organic principles. The vitamin was a molecule governed by the same rules of bonding and reactivity as any compound synthesized on a benchtop. Life's chemistry was chemistry, full stop. The book's influence became immense and global. Its clarity, comprehensiveness, and modern perspective made it the indispensable reference for advanced study. It went through 13 editions under Kerr's meticulous authorship, each one updated to reflect the latest discoveries, many of them from his own laboratory, translated into 7 languages, including English, Spanish, French, Italian, and Japanese. The Kerr became a universal language for chemical education. A graduate student in Zurich, a researcher in Tokyo, or a professor in Boston during the 1940s or 1950s would almost certainly have a copy on their shelf. It standardized the curriculum of advanced organic chemistry and biochemistry across the world, creating a shared intellectual foundation for the field's subsequent flourishing. The direct impact on those who followed is difficult to overstate. At the ETH Zurich, where Kerr taught, his textbook was the central text. Future Nobel laureates like Vladimir Prelog and brilliant synthetic chemists like Albert Eschenmoser were educated within the intellectual framework Kerr had built. Their own monumental work in stereochemistry and the synthesis of complex natural products, including the formidable vitamin B12, grew from the soil he had so carefully prepared. He equipped them, and thousands like them, with a mental map of a vast and intricate territory, empowering them to explore its frontiers with confidence and rigor. His influence extended far beyond a list of discoveries. He shaped the very way the next generation of scientists thought about the molecules of life. In assessing the full measure of Paul Carr's career, one sees the arc of a master builder. He began as an explorer, venturing into the unknown biochemistry of nutrition to find the essential molecular components of vitality. With unparalleled skill in degradation and analysis, he became a brilliant surveyor, determining the precise atomic blueprint of each new structure he encountered, from the long chain of beta-carotene to the intricate rings of riboflavin. His work in synthesis was that of a master craftsman, proving beyond all doubt the correctness of his blueprints by constructing the molecules themselves, atom by atom. His ultimate contribution, however, transcends any single discovery or synthesis. Paul Carr was the great structural architect of early 20th century biochemistry. He revealed that the vital functions of organisms, the processes of sight, growth, and metabolism, were underpinned by molecules of breathtaking elegance and comprehensible design. Through his investigations, he established a fundamental truth that the language of structural organic chemistry was the language of life itself, and through his language, he taught that language to the world. His legacy is therefore twofold. It is written in the very molecules that form the bedrock of modern medicine and nutritional science. It is also embedded in the intellectual structure of chemistry itself, a testament to a mind that found the deepest order within the beautiful complexity of the living world.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.